A very good morning to everyone on this beautiful Friday morning in Mumbai. We are here and I welcome everyone present here on the 25th Somaiya public lecture. I give a special thanks to not only the houseful audience that we have over here, but students who are sitting in the aisle and many more who are joining us live on streaming. And to embark the onset of this auspicious event, I request Ms. Sunita Gangrade from Maya Somaiya School of Performing Arts to kindly recite our campus prayer.
पूर्णमदः पूर्णमिदं पूर्णात् पूर्णमुदच्यते पूर्णस्य पूर्णमादाय पूर्णमेवावशिष्यते ॐ ईशावास्यमिदं सर्वं यत्किञ्च जगत्यां जगत् तेन त्यक्तेन भुञ्जिथा मा गृधः कस्य स्विद्धनम् मूकं करोति वाचालं पङ्गुं लङ्घयते गिरिं यत्कृपात्वमहं वन्दे परमानन्द माधवम् ॐ प्रणो देवी सरस्वती वाजेभिर्वाजिनीवती धीनामवित्र्यवतु ॐ चोदयित्री सूनृतानां चेतन्ती सुमतीनां यज्ञं दधे सरस्वती ॐ असतो मा सद्गमय तमसो मा ज्योतिर्गमय मृत्योर्मामृतं गमय ॐ सहनाववतु ॐ सहनाववतु सहनीवृस्तु सह वीर्यं करवावहे तेजस्विनावधितमस्तु मा विद्विषावहे सर्वैत्र सुखिनः सन्तु सर्वे सन्तु निरामया सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखमाप्नुयात् ॐ शान्तिः शान्तिः शान्तिः
Thank you, Ms. Sunita, for setting a melodious and calming tone to this event. I now request Dr. Achala Danayat, Dean Research and Director of Somaiya Institute for Research and Consultancy, to kindly welcome the audience, our guests, and give a brief introduction and overview of Somaiya Public Lectures. Welcome, ma'am.
Distinguished guests, Dr. Mani, Dr. Massimo, respected Chancellor Shri Samir Somaiya, respected Vice Chancellor Professor Pillai, Secretary Somaiya Trust Lieutenant General Jagbir Singh, Dr. Sangeeta Shrivastava, Executive Director Godavari Biorefineries, Deans and Heads of Institutes, my colleagues from Somaiya Vidya Vihar University, Somaiya Vidya Vihar, Somaiya Ayurvihar and GBL, and my dear students. A very good morning to everyone. I'm delighted to be here in front of you on this special occasion of the Somaiya Public Lecture. And I would like to start with introducing you to this lecture. Somaiya Public Lecture Series is one of our prominent educational outreach programs intended to share scholarly insights on recent trends, emerging areas, and the cutting-edge research, science and technology policy, innovations with the society at large. We bring eminent teachers, researchers, policymakers, educationists, scientists, writers, jurists, innovators, and social workers to offer an insider's view on their own area of work to the cross-section of society. This activity forms part of the efforts of the Somaiya group of institutions to contribute knowledge, thought leadership, and innovative ideas to the general public, faculty, and students. The program showcases dynamic speakers through a number of community-focused talks across a range of disciplines, encouraging debate on issues of regional and global relevance. The speakers share their knowledge, network with the shareholders and with the stakeholders, and entertain and inspire. We have been fortunate to have several esteemed speakers in the past few years since this lecture series started. For those of you who are new to this university, a little bit about the university. We foster an ecosystem that excels in education, research, and service, a place where knowledge is preserved, disseminated, and new knowledge is created. It is known as much for its science, technology, medicine, engineering, management, social sciences, and commerce programs, as for its programs for academic studies in various faiths and cultures of India. With 8 decades of providing enriching learning experiences driven by our motto, Jnana Deva Tu Kaivalyam, we became a university with freedom of possibilities on the 26th of August 2019. Somaiya Vidya Vihar University is the first private university in Mumbai via the Maharashtra Self-Financed Universities Act 2013. We offer varied programs, several undergraduate, postgraduate, PhD, and several certificate and diploma under 9 faculties. So you can see the vast breadth and diversity in the programs that this university offers. As a Dean of Research and Director of Somaiya Institute for Research and Consultancy, I'm proud to host today's event and would like to share a glimpse of our activities. Somaiya Institute for Research and Consultancy, also known as SIRAC, functions as a think tank and a catalyst. We nucleate research ideas, we foster a research culture, and we promote interdisciplinary research across various domains within Somaiya ecosystem, encompassing higher education institutes as well as schools of Somaiya Vidya Vihar and Ayurvihar. The main activities of SIRAC involve the following: research promotion, both internal and external events, such as organizing these public lectures, which is the brainchild of our esteemed Professor Pillai, our Vice Chancellor, annual research meetings and expert talks, research, administration and management. This includes centralized PhD admissions for the university, creating awareness about intellectual property rights, filing patents, as well as offering seed grants to the young research faculty who have joined our university. We also we have research centers in interdisciplinary areas. I'm very proud to share that recently we inaugurated a Center of Excellence in Analytical Sciences headed by Dr. Padma Vankar, jointly with Shimadzu from Japan. We are actively involved in research in the areas of biological sciences led by Dr. Moshmi, and in the areas of regenerative agriculture and sustainable climate action led by Dr. Parvati. With this, I complete my brief about Somaiya University, the Somaiya public lectures, and about CIRAC. During the next 2 hours, we shall delve deeper into one of the most pressing challenges of our times, a challenge that touches the lives of millions worldwide. Cancer, with its complexities and profound impact on individuals, demands our unwavering attention. It is a disease that knows no boundaries, no distinction of age, gender, race, or socio-economic status. Here in the halls of this university, we have the privilege to engage with distinguished experts who, with their relentless pursuit and research in the field of cancer biology, will unravel its mysteries and shed light on the latest advancements in this field. And with this, also instill in us the hope for a brighter future. Thank you, Dr. Mani and Dr. Massimo, for honoring us with your esteemed presence in this beautiful green campus. Thank you.
Thank you, ma'am, for introducing and welcoming our diverse set of audience and presenting the objectives of Somaiya Public Lecture and Somaiya Vidya Vihar University. I would now request Honorable Chancellor Shri Samir Somaiya sir to kindly present a brief address for this event. Welcome, sir. Yes.
Good morning to you all, and to our Italian-American guest, buongiorno. Very nice to have everyone here. I'd like to quote from our prayer, which was sung, Asatoma Sadgamaya Tamasoma Jyotirgamaya Mrityorma Amritangamaya, from darkness to light, from untruth to truth, and from death to immortality. In this case, what we're trying to say is, let us go to knowledge, and our motto also is Jnana Devatu Kaivalyam. So knowledge alone liberates. In this case, we both have Dr. Massimo and Dr. Mani at the forefront of discovery of how to deal with cancer. And it is wonderful to have them talk. They're both at the cutting edge of their fields. And sir, in the prayer we had Sarvetra Sukhina Santu Sarve Santu Niramaya, which means let everybody be happy. Sarve Santu Niramaya is let everybody be free of disease. Yeah. Sarve Bhadrani Pashyantu, let everybody see nice things and let nobody be unhappy. That's the prayer. So, you know, we are working here at the forefront discovery. The dream always has been, how does this academic institution, today a combination of Vidya Vihar University, Ayur Vihar, work together to create discovery of knowledge, science, and create an ecosystem. Many years ago, Mani and I met, and Mani was a postdoc at MIT, dreaming of creating a new molecule to deal with stem cell science in cancer, and he was at the forefront of discovering about metastasis. And we thought that as a company, we would try to create a molecule. Indian companies are very good at creating generics, which is wonderful. But we thought, why don't we try and imagine that we can also create a molecule that could be a blockbuster? And so we started working 17 years ago, and today that molecule is in the clinical trials. So it takes a long time, and the Godavari team is here to try to make a lot of this happen. And also the idea is that it should not only be two of us to do this, the whole country can also have lots of people who dream to do this. and that's the ecosystem we want to create. I want to end, I don't want to talk too much, we want to hear him, but I want to tell a story of money which I heard today. Money came from a small village in Tamil Nadu. His parents were illiterate, am I right? No, couldn't read or write. He was finished his 6th standard, needed 200 rupees to do the 7th. He didn't have 200 rupees to study in the 7th. So his grandmother, who owned 5 goats, he gave his uncle 1 goat to sell. That goat was sold for 200 rupees, and he finished further 7th standard, and today of course he's leading transnational medicine at Brown University in the US. It's incredible what you know, it's an incredible story, and there are many more stories. It's great to meet Dr. Massimo. I just met him yesterday, and he talked about the liquid biopsy so that we can see whether cancer is there in the bloodstream. So these are people who are at the forefront of science. Great for you to be here, and thank you for coming. Welcome.
Thank you so much, sir. I would now also request Honorable Vice Chancellor, Sir Professor Pillai, and our guest speakers for the day, Dr. Sendurai Mani and Dr. Massimo Cristofanilli, to kindly proceed on the stage as we felicitate our speakers. I once again welcome everyone, and as we proceed with our session further, I would request Dr. Amit Gupta to kindly come over the stage and introduce our first speaker of the day.
Yeah. Good morning, everyone. So first of all, I would like to express my heartfelt gratitude to the Dean of SIRAC and Sri Somaiya sir for giving me an opportunity to introduce such a big faculty. That is, being a surgical oncologist, it is always a dream of how the cancer research is going on and how we can take initiatives to curb it down. That is very important. So yeah, and thank you all the dignitaries who are there, respected teachers, friends, colleagues, and students. So I would like, here I feel lots of MBBS students are there. How many of you are interested in research? Can you please raise your hand? So I get insights like for this lecture. Yeah, great. Research in oncology? Good. So many of the people are interested, so it is an important session for you all. So I think sir has started bit introduction of Mani sir, but I would like continuing it further. So I think it's a story of a boy who was from a remote village in southern part of India. He started his journey from there, and he was born to, so parents were farmers, obviously, and then he had a quest for knowledge. He wanted to do something different. That is where your research mind starts working, curiosity. That is where he started his journey of master's bachelor degree from Madurai University. And after that, he got an opportunity to be in Indian Institute of Science, that is a research science, to do his PhD in biochemistry and molecular biology. So that was one breakthrough where he has his mentor as Professor Govind Rajan. So Govind Rajan Padmanabhan is a great figure. He is a Padma Shri, Padma Bhushan. Why I'm telling you, you need to understand when you come in contact with mentors, that is where you grow. So that is this place Sir has formed where you get the best of the best people in the world to guide you. So, and why he was Padma Shri and Padma Bhushan, that time malaria was a very important problem, if I'm not wrong, sir. He discovered, so curcumin was one of the thing which works against malarial thing, which he worked on. And I think that time it was a breakthrough because we had lots of mortality because of malaria that time. So now the things are different because of the research. And that still Sir's quest for curiosity and further knowledge continued, and then where he went to US to complete his PhD from Nebraska Medical Center. And in MIT, he got the, I will say, if you know Professor Winberg, so he is the, if you have read books, I think MBBS, if you go, you hallmarks of cancer. So I think the gene, oncogene and tumor suppressor gene, RAS gene and retinoblastoma, it was his discovery. And that is where Sir started his journey in molecular biology, in oncology. And with Sir's team being there, and he was in the MD Anderson as a faculty also, and later on became a professor. So you should know this MD Anderson, Nebraska, MIT, they are the topmost institutes. Why I'm telling you this all important words, you have to reach somewhere, you have to be at the top level with top mentors, which you are there, and you are lucky to be here to listen to such a great individual personality. So there he started his journey in the lab, and that is where they EMT, so it is epithelial mesenchymal transition. That is where the breakthrough happened, and sir, it is, I will say, it is his efforts where that breakthrough happened, and that was the story of cancer metastasis, that is high. So epithelial mesenchymal is like you all people are in harmony here, and suddenly the harmony goes away. So that is where from epithelial you become, epithelial is an organized form, mesenchymal is disorganized, and that is where cells start spreading. It's a layman term which I'm telling you, sir will tell you in detail. So that was the breakthrough how the cancer metastasis happen. Before that nobody knew how they are happening, why this biology is like that, how to have drugs against that. So if you don't know the mechanism, you cannot do a research and you cannot have drugs against that. So that was one golden breakthrough which he had, and he further continued his journey. He had his lab research, again EMT, and then the other discovery was groundbreaking discovery for Sir, cancer stem cells. So cancer stem cells, that is where EMT is the main component where the cancer stem cells are formed, and that from there they go to blood, because the cancer stem cells is like a multipotent cell. Na? All the cells can be divided from the cancer stem cells. So cancer has its own blood supply. Before we used to think from where the cancer cells are taking this blood supply stream. So to your knowledge, they can produce stem cells from the EMT transition, and that is where they start growing and the metastasis happen. And so same journey continued, research against that. He opened his lab, so that was a research, and after that his journey continued to make more scientists. So now in his lab he has got 40 scientists. One more important thing, this research which Sir made had 8,000 citations. So for you, meaning of citations is, see, whenever we do some research, how many people are reading it? It is a very important component to understand that index. So we have an H index also. So 8,000 citations means a lot, and it is in worldwide, it is one of the, if you say 8,000, very few people have achieved that. So excellent discovery. And then he trained, he has 40 scientists under him, and he is training them for further how to work on the cancer and research and drugs and everything. So and other thing very important, being from India and went to America, so we say as Indian American scientist, but he has made our country proud. He was the first one when he got the doctorate. His parents, he made his parents proud in that area in southern part. He was the first doctorate that time. Nobody was educated so much. So I think from that time journey, embarking his journey with curiosity and passion, he has unraveled many of the mysteries, And being in oncology, I truly understand what does that discovery mean, and which is for your student also it is important, because one of you, if you reach there, you have got people, peers to help you. And thank you very much, sir. So I would like the audience to please stand and give him a big round of applause for his efforts. He has really done a great work, and for Somaiya sir also introducing such people to us, so where we get motivated. Thank you very much.
Battery low. Yes. How about now? All right. Uh, all right. I just want to say thanks to Samir. Leaders, and you see leaders who just give you advice. There are some leaders not just to follow it, sorry, not just give you advice, but just follow it. Maybe I'll stand here. Looks like a speak loud. So, you want me to hold that? Whichever works. This works? All right. Right. So I am so honored, so thrilled, and so happy that I got a chance to meet Samir at some point in my life, who literally, made me realize the importance of being a human being, right? Not necessarily, success is something which comes and goes, but human being remain human being. And that's a lesson I learned from Samir. And he has been inspirational ever since. I just follow him on many aspects. Particularly, it's very important to be simple and humble human being, no matter who you are. Okay, now coming back to science. So the way I designed my talk, such that it should be simple, but at the same time, if any of you want to ask questions, please feel free to stop and ask me. And we can make it as an educational and rather than just a lecture, right? So why do I care to study, right? Like you are introducing the topic. Cancer is one of the major problem which we all need to understand. Not because you want to make a next breakthrough, but it's personal. For many people, it's personal. It's important to understand so that we take a proper precaution, even to protect ourselves, our family, our friends. So it's vital to know for that reason more than whether I am going to be a next future scientist or not. I hope you all will be, but independent of that, just pay attention to the lecture and feel free to ask me. I'm here
to give you the reason and purpose of studying this particular disease. So this slide shows cancer incidence and cancer mortality in India today, or 2022, which is on the left. I have a mouse. Okay, now. See, on the left, in 2022, it's 1.41 million incidents, which is expected to go to almost 2.7 million by 2050, right? Now look on the right, which is a cancer-related death. Today, in 2022, it's 917,000, which is expected to go to 1.83 million by 2050. It's based on calculation, statistics. So this is in the US. And you see in 2022, the cancer incidence is 2.38 million. It's expected to go to 3.5 million. The cancer-related death is 606,000. In 2050, it's expected to go to 975,000. Okay, it's a data. I just put them all together in one slide, just for you to get an idea. 1.41 million, 2.3 million. Out of 1.41 million, 917,000 gave up their life, versus 2.38 million, which is almost more than 1.5 times, but it's only 606,000, which is, I'm very thrilled. I'm there in the US. We are able to witness the success of medicine. Okay, 5 years ago, somebody had a breast cancer. It was a death sentence. But today, more than 90% of the people can survive, because thanks to modern medicine. I did not, Suthi. I was just curious to know what is the, thank you. So, but the point I'm trying to say here is that it's important for us to research, understand, prevent. So in the future, you know, this data, what you see here, this is expected to be 3.5 million, and expected death is 975,000 approximately. And here we are thinking 2.7 million incidents with 1.83 million cancer-related mortality, which means we need to work hard, spread news, prevention, awareness, more treatment, more medicine, all that need to happen here. So one of the area we study in my lab is called cancer metastasis. Why do we care about cancer metastasis? So this slide shows, for example, in case of prostate cancer, as well as in breast cancer, if they have a localized disease, which means the tumor is present only in the site where they form, the survival rate is very high today. This is a data from the US. But if the tumor is metastasized, the survival rate drops to almost 25%, right? Which means if you can find a way to stop metastasis, you can let people live longer than today. So what is metastasis? It's a very complex process where a tumor forms at a site, let's say breast tumor, forms in the breast site, and then at some point, the tumor recruits more vasculature, a blood vessel, and now, because once the tumor grows beyond a particular size, they cannot get enough oxygen, so that's when they trigger something called hypoxia. Through that, they bring new blood vessel into the tumor, and these vasculature tend to be leaky sometime, or sometimes the tumor cell become very aggressive. They find a way to enter into the blood vessel. This process we call intravasation. Once get into the blood, it travels and get out at the newer site, and they create a tumor at the new site. And this process is called a metastasis. In 1999, when I started my research, there was very little was known about the whole cascade. We knew this is happening, but we did not know how. That's when I got started. I asked this question. So we have written a review article. In case any of you want to read, you can go to this review. You get a lot of information. It's summarized in detail. Let me just give me a second. So I'm going to talk the biology behind how this happens. But before that, just want to give you what do we do today? What are we studying right now? Okay. So here we all have something called immune cells in the body. Okay. These immune cells at the beginning of this tumor biology, what it does, it stops the tumor from forming. We call that as a tumor killing immune microenvironment. Sorry, it's a here. We call that as a tumor killing immune microenvironment. But at some point, the tumor switches the microenvironment from tumor killing to immune suppressive. Okay, tumor killing to immunosuppressive. At that point, the tumor is highly aggressive. Okay. So we call this tumor or immune microenvironment change from a cold to hot or hot to cold. So I will explain in a minute what I mean by cold and hot. So if a tumor has a lot of immune cells which are able to kill the tumor cells, they are called hot tumor. It's not temperature hot, it's got a more immune cells to kill the tumor cells. At some point over a period of this metastasis or tumor progression, the tumor switches the immune microenvironment from hot to cold, which means they somehow either kill off the immune cells which come into the microenvironment or just make them inactive. I will show you in the next slide. So here you can see the so here we have labeled the tumor cells in red and the immune cells or the T cells in blue. One second, I think this is a very important video I want to show. Yeah, easy. So the T cell, which is in blue, is trying to kill the tumor cell, which is in red. If the T cell is able to kill the tumor cell will turn green. You can see that beautifully on the left. But we do the same thing from a metastatic tumor. Okay? Here is a T cell is interacting with the tumor cell, trying to kill the tumor cell, and you see it try. At the end, the T cell die, not the tumor cell. Okay? So, and we have tested, and on the right you can see in the bar graph that among the T cell killing, there's only 17% of them from this non-metastatic tumor is able to kill the tumor cells. But from a metastatic tumor, not even a single T cell was able to kill a tumor cell. Okay? So we kind of did a thorough research and we figured out that it's because the tumor is telling the immune cells that I'm your friend, don't kill me, and at the end, the immune cells don't kill. So we are trying to figure out how to make these tumor which are otherwise cold into hot tumor, right? If you can make them hot tumor, most of the treatment will work, including chemotherapy. So that's one of the major effort in the lab. And these students, Brianna was a PhD student, along with these other two students, they are trying to do that. So I think my slides are a little bit out of order, but I will explain. So you are talking about, when you are introducing, you're talking about mentor. So mentor play a vital role in our life. So when you see a mentor, right away you should figure out a way to get help from your mentor to not just intellectual knowledge, life knowledge, right? So I was fortunate many years ago to work with somebody with the name Robert Weinberg, who's a National Academy member. So he was very simple person. So when I joined his lab, all he said was, ask crazy scientific questions to solve important clinical problems and find solution through systematic research. That's it. He did not tell me what I should work on, and that's all my guidance. But he created an environment where you can find what you want. So I asked this question, how tumor develops to become metastasis? As I said earlier, there was very little was known in 1999. When I joined, at that time, the Weinberg lab, they published a paper where they were able to get a breast epithelial cell from a normal individual and then put some of the genes that it case shown. This is it's called ASP40, early region, telomerase, and HRAS, some genes. You put them. Each one does different things, but it makes a cancer. Okay? When you put these cells in mice, they form a tumor. And the person who did the work, he said that I'm able to, that was the first ever discovery of somebody making a cancer cell in a lab, not by exposing to chemicals, but by manipulating genes. So why is it different? So when you throw a chemical to a cell and convert a normal cell into a cancer cell, you have no clue what happened inside the cell. It could be 1,000 things would have happened, or it could be only 2 things would have happened to make that normal cell into a cancer cell. But here is the first demonstration ever that just by manipulating these 3 genes or some pathways, you can make normal cell into cancer cell. When you put these cells in mice, again, they form a tumor, but they go nowhere. They just stay where you inject. So he was happy. He made a landmark discovery. Now he's a director of something at Dana-Farber. He's a big shot, a clinician, scientist. But that's when I started asking this question, why? Why are they not going anywhere? Can I find out? So I partnered with another postdoc at the time in the lab. We asked this question, and we found out. I'm not going to tell you exactly how we did all that. I'll be happy to share that if you're interested. What we found is that if you take a normal breast tissue, okay, if you see on the left, they are epithelial looking, they are highly organized, and when these cells get transformed to become cancerous, they still bound with one another through something called cell-cell adhesion molecules. So which you see here on the slide, that they are attached with one another through various cell-cell adhesion molecules. Okay? So that is why, because the tumor cells are bound with one another, they don't go anywhere. They just to get transformed, become cancer, they just stay where they are, where they formed a tumor. So what I found at that time, again through various systematic approach, that they need to break this cell-cell adhesion, and they need to break the cell matrix interaction, and which is shown in the red. You see that cell-cell adhesion, that's a cell matrix interaction, second one. That process we call epithelial to mesenchymal transition. Why do we call epithelial mesenchymal transition? When they break this cell-cell adhesion, they become migratory mesenchymal, or they are more motile. And this happens during embryo development. The first cell forms following fertilization is an epiblast. That cell develops into this EMT program and creates a mesodome. Mesodome gives rise to heart, bone, your blood cells, and all that. So this process happens during embryo development. Cancer steals that program, and through this, it's able to make the tumor cells travel from one place to another, okay, and develop metastasis. So again, one of the most important thing I would say as a student, I always do, I would recommend those of you interested to do this. I always do strength, weakness, opportunities, and threats. So what it means is SWOT analysis. What it means is that you ask yourself how good you are in what you are good at, and nobody is good in everything, right? We are all good in something, and there are others who are good in something else. When you need something, you go and approach somebody else where you are not good at. It's okay. Take their help, acknowledge them, appreciate them, and if you give a right credit, they will be fine. Like this, one day, I was giving a lecture at the Whitehead Institute, not I was giving, somebody else was giving a lecture at the Whitehead Institute. I was sitting in the auditorium. I was faced with a major problem at that time, which was when a cell leave the primary tumor, they go and develop metastasis. The metastases tend to look like where they come from. Okay? If somebody has a metastasis in a lung, a pathologist could look at a tumor and they can say this tumor comes from breast, prostate, lung, liver, spleen, just by looking at it, right? I was asking why, how, right? Does EMT makes stem cells to the, or makes cancer cells to become stem cells? You know, when a stem cell go to a newer site, they can create an organ similar to where they come from. Very simple, okay? Very simple hypothesis. Again, I was good in doing a lab work, but I was not expert in stem cells. I approached this guy, Maijing Leo. I said, Maijing, I wanted to test this idea. He was expert in stem cells. He said, it's a crazy idea, but doesn't take much time to do just a simple facts analysis. Let's try. So he did. Long story short, that my hypothesis was right, that through this EMT program, sorry, sometimes the pointer works. So through this EMT program, there you go. I guess this is switching between two screens. Sorry. All right. So through this EMT program, now cancer cells, can you see my mouse there? No? It was visible? Yeah, there you go. So through this EMT program, cancer cells not only become mesenchymal and migratory, but also become stem cells. That transformed my life, made a huge impact in cancer biology. Why? So cancer stem cells are responsible for metastasis, chemoresistance, and tumor relapse. Okay? As you were mentioning earlier, this paper now has more than 9,500 citations. So when I went to the Weinberg lab, I was thinking, will I be able to survive in his lab? And everyone in his lab, I went from Indian Institute of Science, Bangalore. So I'm joining MIT, Whitehead Institute, Robert Weinberg lab, and everyone is like an unbelievably smart people. And question I wanted was, can I survive? I wanted to survive, not be successful, survive. But at the end, today, among all the paper Bob Weinberg published in his whole career, my paper ranks #1 based on citation. So thank you. But the reason I'm saying is that more than this, you know, it's very important to be persistent. Thank you so much. Yeah, persistence makes huge impact. So it took, people normally do 3-year postdoc. I did 8-year postdoc, and it was very hard. But at the end, it became meaningful or fruitful. So why do we care? As I said, in a tumor, you do have stem cells. Now, if you give a regular treatment, that will eliminate most of the cancer cells, but stem cells will remain. Those stem cells can differentiate, proliferate. They can develop a tumor again. That tumor could also be metastatic, right? So if you have a cancer stem cell specific drug, you can eliminate the cancer stem cells, but the remaining cells can undergo dedifferentiation through this EMT program I was talking about. The tumor will relapse. So the ideal scenario would be combining a drug which targets cancer stem cells and the differentiated cells. So which we are working. For example, the company which Massimo was referring to is trying to develop drugs against cancer stem cells. And we have a number of standard of care drug with the goal of one day we will be able to combine and then eliminate tumors, which we are working towards. We have a number of drugs in the lab. In the interest of time, I'm not going to cover that. There are a number of people in my group, they are trying to understand or develop new drugs to target cancer stem cells. So another problem we are approaching in the lab is that you cannot see here, tumor is not just one type of cell. Tumor has many different type of cells. Each cell has a unique its own genome. Okay? It's very important to understand the complexity of these tumor cells. I will explain in a few slides. By understanding that, you can understand how the two cells communicate with one another. Okay? if you can break the communication, you can literally get rid of the tumor. So that's what most of the drugs today we have are trying to break the communication between two cells, or making the immune cells to be awake so it can go and kill the tumor cells, a number of things. So in our lab, Massimo is going to talk about liquid biopsy, which is kind of looking at how the tumor cells, when they are in a blood, how unique they are or how different from one another, and can we capture that, which is kind of his finding revolutionized cancer diagnosis and cancer care at a precision personalized level. He's going to talk about it later. And again, by the way, he was the first one to ever demonstrate or start a personalized precision oncology clinic at MD Anderson, and he was the first one to show something called a circulating tumor cell, which has a clinical relevance, which is widely used today. So in addition to figuring out within a primary tumor, can we differentiate the tumor cells from one another, we are also looking at when a stem cell divide. Here is a marker, we use something called a FOXC2, which is a protein. Here is the two daughter cells which are dividing, and you can see the nuclei, and both the daughter cells is marked here, but only one daughter cell has this protein, not the other daughter cell, which means at the time of cell division, this is literally 24-hour time period. Okay, in that there is a 3-hour window when the nucleus is divided. Now the cell is going to decide, am I going to be a stem cell? Am I going to be a differentiated cell? Just imagine it has got a 20,000 plus genes, and in a 2-hour time window, it's deciding the fate of the daughter cell. How the fate is decided, right? One can say, yeah, it's deciding, who cares? But understanding that is going to help us. How can I stop the cell from making a stem cells, right? If I can stop that, it's huge. And that window is just a 2-hour window in that time frame. So that's something we are in the process of asking in the lab. So today, a patient walks into the clinic. I wouldn't say today. Before Massimo thought about the precision oncology. When a patient walks into the clinic, you give a standard treatment, and the patients are not uniform. Each patient is unique. But you give a one care, standard of care treatment, and you expect miracle for every patient. Doesn't happen. For some patient, it has a very good effect. For some patient, the drug has no effect. For some patients, you have a lot of side effects, right? It's extremely important to classify the patients and have a right drug, okay? Thanks to genetic profiling, we are able to classify the patients into different groups, and now we have a drug for the individual patient's genetic profile, okay? And you will hear more from Massimo how they are able to use this liquid biopsy to separate patients into different groups. And now if you have a drug for each group of patients, you get a very good effect. That's what we want, right? That's what we are working towards. It's called precision medicine. Again, as I said, the concept was long ago started, and Massimo was the first one to kind of establish this clinic for personalized precision medicine. So how is it possible? If you think about profiling patients, when I started a postdoc again at the Whitehead Institute at MIT, Eric Lander was trying to sequence the human genome. He spent $3 billion almost to sequence one genome, and they spent a couple of years to sequence one genome. Today, we can sequence it in a day, and we can sequence it under $1,000. It's going to go down even further. Maybe in 5 years down the line, all of you in your iPhone, you will have your genomic profile, and you will be walking around with it. So that's where we are going to. So that really helped to stratify the patient population into different groups. Here is the one miracle example. This is very old data, but this patient had a mutation in one gene called a BRAF. It's a melanoma patient. He's got to look at the number of tumor this patient had. 15 weeks of treatment against using a drug against this mutation, BRAF, the same patient, right? So that's a precision medicine. And this is one example. Now, we have a drug for multiple mutations, and every day, every month, there is a new drug getting approved for these mutations. So, what I foresee to see, there is a name ILAN written, I will explain what it is. So, ideally, we have a number of omics, it's called methylome, genome, proteome, RNAome, there's all kinds of omes, right, all omes. And you want to combine all of them and then get an idea who's that patient, right? And we feed that information into a supercomputer. I'm just telling the future. This is what it's going to look like, okay? We feed that information into some cloud-based computing, and you get an information about more effective, less toxic, individualized treatment plan. It's going to happen 5 years, maybe less. But today, what do we do? So I came up with an idea in collaboration with a colleague here. Her name is Seventhi Limaye. We thought, why didn't we put together experts who are literally those supercomputers? Massimo is one among those. And then we feed this information to them. We'll ask them to spit out some information which would be beneficial for patients. And that's the way we created a company called ILAN Precision Oncology, which is serving now cancer patients around the world, okay? Now, that's the good news. But again, the problem comes, right? While this patient responded well to this treatment, the tumor came back. So that's a problem. It's a tumor relapse. So the tumor is always finding a way to escape. Once they develop a mutation, once they become a tumorigenic, they find a way to escape and develop a tumor again. So you need to understand the escape mechanism, how the tumor is escaping. That's something we are in the process of asking in the lab. For example, one could argue the primary tumor had those resistant cells. It pre-existed. You gave a treatment to the BRAF inhibitor. You got rid of all the BRAF positive cells, but there are tumor cells which are not BRAF dependent, but they depend on something else. But they pre-existed, and during the treatment, you eliminated most of the cancer cells. The remaining cells which you did not eliminate just took over the tumor. We call clonal expansion, right? These clones pre-existed. Now, the second idea is these cells did not exist before. Rather, during the treatment, in response to treatment, tumor wants to evolve, constantly wants to evolve. In response to treatment, they gain new mutation. They create new cells. Now, those cells expand and create a tumor. This is called acquired resistance or clonal evolution. Okay? They evolve these cells. So we are trying to ask what is happening in a patient. For example, here is a study we are in the process of doing where a patients walk into the clinic. We collect the tissue and blood from this patient, and this patient's going to go through the treatment for one of the drug called a RET inhibitor or any inhibitor for that example. Then you collect the biopsy over a period of time, and then surgery you can study over a period of time whether the tumor cells are evolving or tumor cells are getting selected by looking at this profile. Again, why is it important? You know, when a pathologist look at a tumor, they just look at it like this. There are so many cells inside. What are they? Who are they? And why one cell is sitting next to one another, right? You can ask why Samir is sitting next to Massimo. If Samir is saying something to Massimo, only they can hear. You cannot hear. So the communication, it's very important. Who is sitting next to you is going to make a huge impact that what information you hear from your neighbor. You cannot hear from me unless otherwise I'm using a mic. So it's vital for us to understand all these information, the complexity, and that would help us to develop new medicine. In my lab, I just moved to Brown University. I'm the associate director for a translational oncology at Brown. I got a big startup money. So what we did as soon as we moved, we purchased this machine. It's called a Chromium. It's called a Visium, another one. This is called a Xenium. They are more than $500,000. But the key is this one and this one can map every single cell in a tumor. Okay? You can characterize them at a single cell level. This one just to characterize them at a single cell level. This one will give a spatial context. It'll say this single cell was sitting next to that cell. that single cell was sitting next to this cell. So once we know that, this is through barcoding technology. You're welcome to ask me if you want to know more about it. I'll be happy to share. Okay, it's called a barcoding. Each cell is barcoded, and then we characterize them. Once you get that information, this instrument is the most expensive one. It's it's called a xenium. The people in the lab call it Xena, where you can map all the cells on the slide, even if the tissues are harvested 10 years ago from a patient. So why do we care about tissues harvested 10 years ago, right? Who cares? But the benefit of 10-year-old tissue is that you have a clinical follow-up information for this patient. In other words, we know today what happened to that patient 10 years ago, what kind of treatment that patient received, did the patient live long or how long, all that information we have. So we can feed all that information and then figure out what mutation probably making the patient live long or live short. If we can understand that, it's easy to go back and manipulate those signaling. Each run costs $4,000. That's where the money is, not the instrument. But luckily we have money to do that. Teamwork. Teamwork is one of the key ingredient, in my opinion, for us, for me, for many of my colleagues to be successful. We all work together to figure out a way to understand this complex disease. So here is a slide. This is Jim Allison, who discovered that immune cells are forced to become silent by the tumor cells. If I can go back and manipulate that, I can make the immune cells kill the tumor cells. People laughed at him, right? It took many years for him to prove to the people that he's right. He got a Nobel Prize. It changed. Now, melanoma 4 years ago was a death sentence. Today, significant portion of melanoma patients, they feel the benefit of immunotherapy. And it's going in every other cancer. So we are looking in breast cancer, right? It works, it doesn't work, why, how, all that, right? Understanding that, it's a very, very important thing. Massimo Cristofanilli, my good friend, right? Again, this is a picture from China, right? This is from China when we went to China together. So, Massimo, he has been preaching the diagnosis, right, is as important as therapeutic research. Right? Many people say, if I have a good drug, I can cure cancer. Wrong. You need to have a good drug and you need to have a right diagnosis. So he goes all the time preaching precision diagnosis with a precision medicine is going to cure cancer. We are going to get there. So this is some review article we have written in my lab asking how the field is evolving in the space. Philip Sharp, he's a Nobel laureate as well. He's an amazing guy. He always says science should be treated like a hobby rather than a profession, right? If you look at science as a way, I want to understand, let it be cooking a meal or let it be discovering how EMT becomes, through EMT cancer cell becoming stem cells. If you just be curious, just take it as a hobby, you will be able to make a huge discoveries. Again, another thing we do in my lab is training the future generation. Again, a lot of people got trained and moving along. Here are my lab. This is like 3 months ago. Now I have many more people. Here is the funding support. At the end, for me, these are the guys who teach me the most important thing in life, right? Be grounded. It's very important that you are just another human being, right? Nothing special. The future is bright, okay? And it's very important. It's not far. We just need to work towards it, okay? And again, I thank all of you, especially Samir and rest of you for giving the opportunity. Any questions for anyone? Don't feel shy. It's fine. Yeah, go for it. Yes. What it means, cell barcoding? So, um, you know, let's say there are 500 people sitting in this room. Okay, there are 500 people in this room. Huh. Now, what you can do, you can just simply, how do you map each one is where each one is, right? I want to know where each one is sitting in this room, and I want to know each one's names. Each one of us have our own name, so we can figure out if I can write down and I know exactly where you are, which seat number and all that, right? In a barcoding, we take a small piece of DNA, we know what the DNA sequence is. We attach to you, we give a name for you. The name is a 5-letter nucleotide code, okay? And just to randomize 5 nucleotide, even a 6 nucleotide, randomize it. What is the probability? How many different combination you can make, right? So each one is attached with a 6 nucleotide randomized. So you can have A, T, G, C, T, G, another one has, so that is the code. So we attach that to each cell, okay? Once you attach, then it binds to it. Now you take each cell, we sequence it, then once you sequence it, based on the barcode, I can say this cell had, based on the barcode, I can say this is that cell, this is that cell, this is that cell. So basically giving a name to each cell using a 6 to 7 nucleotide DNA attaching to the cell. We have a technology to attach, so that's easy. Now, make sense?
Yes, sir.
Thank you.
Sir, I had a question here. Sir,
Fine,
I don't know if this treatment exists right now, but theoretically, is it possible that just like we produce antitoxins for venom or like blood transfusion reactions, so one of the mechanisms by which the cancer cells evades the immune system is by not our immune system doesn't recognize it as abnormal cells. So, but any kind of cell, normal or abnormal, inside another person's body, my abnormal cells, but inside any other person or any other animal's body will be recognized as foreign cells. So antibodies will be produced against it. So is it possible to isolate those specific cancer antibodies in the bodies of other animals, and then by isolation and then introducing them as a treatment in that specific person?
So it's a good question. So he's asking, can I take a tumor from a patient A, inject to an animal, and then make an antibody, and then use the antibody back to the patient, and will that antibody work? Will that antibody cure? Okay. Right. So it's a, so for example, there are a number of antibodies which are designed for a particular antigen. So it's very important that you need to have the right antigen, right? So we have an antibody-based therapy, which Massimo will probably talk about it, how the antibody-based therapy works. But it's not as simple as the way you said, take a tumor, inject to an animal, make an antibody inject, because you need to humanize it, right? If you inject them to the animal, it will make an animal-specific antibody, but you need to humanize it. We have a methodology to do it. People are doing it, but it's a cumbersome process, doable, but it's, you know.
I do recognize there are two problems. One is that heterogeneity, that is like we need multiple types of antibodies. So maybe that can be counteracted by the barcoding technology, like individual cells.
So the barcoding technology is useful for research, not for therapeutics, right? Because you don't want to introduce the DNA to a patient, right? So, yeah, good idea. Thank you. So all it does is it generates the data. It's a standalone, but then the data needs to be analyzed, which I cannot. You need a bioinformatic experts who understand, who can kind of analyze the data, give a right meaning. And I have a people in my group, my team has someone who knows how to understand, how to decode, and tell me in a simple term which I can understand. So there is no built-in high-performance computer there. You don't need it. It has its own computer, very simple computer. maybe a little complex, but then our laptop. Yeah. Okay.
Here.
Sorry, am I running out of time? Your time? Okay.
Can I take this question?
Please, please.
Okay. I'm just curious to know, you said that these cells migrate from one tissue and then they go on to another site. What is it that inhibits it to differentiate into the tissue of its origin? And the second is that, is there anything known if or when these cells delaminate from the site, the levels of that protein that is inhibiting it? If anything is known about the levels of that protein within the cell next to the delaminating cell.
Okay, good question. So she's asking, let's say a tumor cell is leaving from, let's say, breast tissue. It went to the bone or it went to the lung. Now it has an option to stay as the cell it left, just sit there, or it can differentiate, because remember this is a stem cell now, it can differentiate and create a tumor. Do we know the mechanism and how it is, if it is? So I said earlier, the tumor cell, they are bound with one another through something called cell-cell adhesion molecules. Now I also said they need to break from one another in order for them to develop metastasis. So what we know today is that the stem cells are important and the cells need not completely break away from one another. Rather, even if they are still has some characteristics of the original cell, they need to gain this mesenchymal cells I was telling you, that attributes. So in other words, imagine if I am a runner and I never run, so I just need to gain a little bit of ability to run, but retaining who I am without me being a complete runner. Okay? So the tumor cells, when they break, instead of breaking completely, they keep that original identity, but put on the mesenchymal attributes. Okay? We call that as a hybrid EMT. Okay? Those are highly aggressive metastatic tumor. Okay? Let's say a tumor cell is completely broken away from one another, they are like a single cells. When they go to new site, the immune cells are saying, no, 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 you should not grow, you should not proliferate, you should just don't do anything. So those cells remain something called a dormancy. In case of breast cancer, for example, the ER positive breast cancer, many patients, they respond beautifully to ER positive treatment. Massimo is the boss and I am not, but I'm just to give you what I know. Okay? When these cells become, when they, in this ER positive breast cancer patients, you treat them, you think that the treatment is working very well, the patient is almost declared cancer free. 10 years later, the tumor pops up. So these tumor cells go to the bone, sit as a single cell for many, many years, and the immune cells keep them under check. At some point, either aging or could be stress, could be many inflammation related phenomenon could trigger these cells to wake up and create a tumor. So one of the things we have been asking with the collaboration with many others, should we leave these cells if they are dormant, we call dormant, remain dormant? Should we kill them while they are dormant, or should we wake them up and when they divide into many cells you kill them? So we have a drug for different things. So those are the ongoing research today to figure out the dormancy phenomenon, which is a very important problem among ER positive breast cancer patients. Okay? All right. No.
So the similarity.
I think we can talk private.
Sure, okay.
Sir, we have one last question here.
Sir,
Okay.
I don't know if I should be asking this question to sir.
Okay.
I don't know if I should asking this question, but sir, what are your views on treating disease and cancer as a whole on the basis of eugenics? Especially because you've been in America, I know that you might have had some idea about eugenics.
You know, it's
It
an
is a bit
evolved.
sensitive topic.
Huh?
It is a bit sensitive topic, I know, but what are your views on treating disease as a whole using eugenics?
I think it's Massimo, do you want to comment? We'll discuss later.
Yes. Thanks.
Thank you.
Thank you so much, sir. As I was sitting there listening to your entire lecture, and I'm noting down points like, wow, this is a good take-home message. And after every 5 minutes, I have another good take-home message, and I'm like, oh my God, how do I summarize this? I think I will just put it together in one sentence. Apart from the wonderful, impactful science that sir has already been speaking about, I don't think I'm the person who has to even tell how great it is. But one important aspect which very beautifully Dr. Mani has amalgamated in his talk is the importance of soft skills, the importance of our environment, the nurturing, culturing environment, and always staying grounded. So I guess that is going to stay with us, and of course, taking science as a hobby rather than as a profession. Thank you very much, sir, for this wonderful session. I'm sure many of you would like to connect with him. Of course, that is possible after the session as well. So moving ahead, I would now call on stage Dr. Maithili Athavale, Assistant General Manager from Sajjan Therapeutics, to introduce our next speaker. And I would also request Dr. Massimo Cristofanilli to kindly come on stage. Welcome, sir.
A very good afternoon to everyone, and I welcome Dr. Massimo to our beautiful country India and beautiful campus of Somaiya Vidya Vihar. It is my privilege to introduce Dr. Massimo. Dr. Massimo has many accomplishments and awards to his bio, but I would like to brief it just in interest of time. Dr. Massimo is an accomplished board-certified medical oncologist with more than 2 decades of experience as physician, investigator, researcher, and leader. He has demonstrated original and innovative vision in the field of molecular diagnostics, liquid biopsy, translational research, and drug development. Additionally, Dr. Cristofanilli has extensive background in clinical trial design and is a key thought leader in the field of metastatic and locally advanced breast cancer. At Weill Cornell Medicine and New York Presbyterian, he served as Director of Breast Medical Oncology, the Associate Director of Precision Oncology at the Mayo Cancer Center, MCC, and co-leader of the MCC Breast Cancer Disease Management Team, as well as the Scientific Director of Englander Institute of Precision Medicine. He has received his medical degree from University La Sapienza in Rome with honors, where he subsequently completed his fellowship in medical oncology. He completed an internal medicine residency at Cabrini Medical Center in New York and a medical oncology fellowship at the University of Texas MD Anderson Cancer Center, where he also served as faculty in the Department of Breast Medical Oncology for more than a decade. He has several leadership positions over the years, including the Chair of Medical Oncology and Associate Director of Clinical Research at the Fox Chase Cancer Center and Associate Director of Translational Research at Thomas Jefferson University. From 2015 to 2021, he was a professor of medicine at the Robert H. Lurie Comprehensive Cancer Center at Northwestern University in Chicago, where he also served as the director of the breast disease team, director of Robert Lurie Cancer Center, OncoSet Precision Medicine Program, and associate director of clinical research. In addition to his clinical expertise, his research focuses on biomarkers of endocrine resistance in breast cancer, liquid biopsies, and novel drug development. His research in the areas of novel drug development, circulating tumor cells, and ctDNA have led to improved treatments for breast cancer. He is an internationally recognized expert in the research and treatment of inflammatory breast cancer, the most aggressive and deadly form of breast cancer. He is the founder and president of IBC International Consortium Additionally, he is globally recognized for his contributions on the detection of micrometastatic disease in breast cancer and a key opinion leader in drug development in hormone receptive positive metastatic breast cancer. He has co-authored more than 400 peer-reviewed manuscripts. With this brief introduction, I now welcome Dr. Massimo to speak on liquid biopsy revolution in breast cancer, tumor heterogeneity, longitudinal monitoring, and cancer interception. Let us welcome him with a great round of applause.
Thank you. That was quite an introduction, certainly not brief. Well, first of all, I want to thank my friend, Dr. Mani, that brought me here. It has been an experience, has been an incredible week. You know, sometimes you go on a trip and you never know what you're going to learn or what you're going to find and how your journey will going to be meeting other people. So first of all, I learned a lot about you that I didn't know, even though we have been friends for a long time. You brought me essentially to meet your family, and it was a privilege. And then I met Samir, and obviously the university, the concept of the university, the new concept of socialized medicine. I think you are a pioneer, and we will need to really recognize you for what you're doing. This is an incredible effort. So my talk actually naturally follows your talk because I am the clinician standing in the clinic and trying to understand what is wrong with my patient, why they don't respond to treatment, in particular why every individual patient is different from another, even though they have the same type of disease. Hormone receptor positive breast cancer, triple negative breast cancer. Every patient though is unique. So we need to learn not only to treat the disease, but actually to treat the patient. But in this process, I asked myself, when I started MD Anderson, an incredible environment, by the way, where you can essentially do everything. I think Manny explained in details. How can you differentiate these patients on the basis of a test, on the basis of something that we can learn, repeat over time? Because I think, for what I see in the clinic, then the disease changes very quickly. So the center of all of that, as you heard, is cancer cells. Now, in the lab, you can work with cell lines and you can look at these cancer cells, but in the clinic, isolating these cells has been extremely difficult, if not impossible. So in 1998, this immunomagnetic separation became possible, was just to be tested. And I brought this company, this machine, to MD Anderson in order to run a clinical trial, a multi-center clinical trial, that eventually revealed us that we can isolate, we can see these cancer cells, and we can count these cancer cells. The simple counting at that time was actually very powerful. And with 177 patients prospectively enrolled with multiple time points, we actually demonstrate that this is a very important prognostic factors. You can identify patients, they have a number of cells, more than 5, where the survival is very short. In a number of patients where less than 5 CTCs, epithelial cells in the blood, in 1 tube of blood, we actually do very well. And so immediately, this was the beginning and not the end. But I think in spite of being so convincing, and I can tell you there is no drug, no treatment that can divide these patients in this particular way. So 177 patients. In spite of this, we had spent 20 years in trying to convince the medical community that CTC are an important factors. So right now we recognize that this was a major milestone and opened the field of liquid biopsy, but we took 20 years trying to explain exactly why is the case. So first of all, back to the observation in the clinic. Why there are patients who have the same imaging, the same disease, but they behave different? So this is an example of 2 patients. They have different behavior. We know that liver metastasis is the worst condition that you can have for a triple negative breast cancer. A triple negative breast cancer with liver metastasis. If we ask any oncologist, they will tell you this is not a disease that we ever respond to anything. Well, in the context of the study, and immediately afterwards, we learn that patients with low CTC or 0 CTC are in fact able to be treated effectively with multiple line of therapy. So their survival is not short. But if you have the same patients, even though at the same imaging, in even a more indolent disease, an ER positive, with a number of CTC, this patient will not survive, will die very quickly, and will not respond to any treatment. So this was the link between the CTC enumeration and the clinical behavior. And I can give you another example. Many times we say the bone metastases are indolent disease. Well, I can challenge you that there are patients with thousands of cells, even bone metastases, and these patients progress very quickly, and they can even respond, but even if they respond, the disease is progressing. So I'm bringing you to the concept of metastasis. This patient progressed to leptomeningeal disease in spite of the fact that the PET scan show an improvement in the bone disease because they started with an extremely high level of CTC. So the decrease of the CTC in the context of a high baseline predicts the development of visceral metastasis. So we spent years trying to prove every single point that we would like to see in the biomarkers. Is the CTC enumeration a predictor of metastatic disease? Yes. Is the CTC better than imaging predicting outcome? Yes. Is the CTC longitudinal count a predictor of benefit from treatment? So all of this actually was done for a concept that is relatively new. 2005, the test was approved, and we had used it in the clinic, and we started to essentially develop a protocol for data collection. So we developed the concept of real-world data back in 2005, and by 2008, we had more than 400 patients for which we can go by retrospectively and address these questions. So one of the questions is, what is the prognostic factors in the context of every single treatment modalities? And these are patients that on the first line, if I have a patient estrogen receptor positive in the first line and start chemotherapy, at that time we use chemotherapy single agent or combination, and we use even bevacizumab, how this patient will behave, how can the CTC really telling us also what type of treatment? And you see that endocrine therapy single agent clearly is not the way to go. These are patients that for which CTC more than 5 predicts for endocrine resistant. At that time, we didn't know what endocrine resistant meant. And so the survival is short, the progression-free survival is short. We were advocating that you should give chemotherapy to this patient. It took really 15 years to run this trial. It proved prospectively that patient with more than 5 CTC, you should get chemotherapy. That study was actually conducted in France. So the second question was, what happened to the patient with more than 5 CTC? If they have the same imaging and they have the same disease to start with, but they have only more 5 CTC, what happened to them? When they progress faster and they progress in other sites, in the visceral metastasis develop. So clearly, is a different behavior. And this links to what Mani was saying. These are cells that are responsible for the metastatic process. These are probably stem cells. So there have been a number of studies actually to look at the characteristic, biological characteristics, the adhesion molecule, the clusters, the CD44 expression. So these are in fact stem cells that we detect with this technology. And in fact, when this study, the first study was validated from almost 2,000 patients prospectively run, looking at the specific behavior of this patient, we actually hypothesized that the CTC tell the truth about metastasis. It's a patient population for which the metastatic process is activated. So the next step is, if we have CTC as a unique biomarker, not simply a measured tumor burden, maybe we should stratify patients in stage 4 disease based on the CTC detection. And so we put together the data from the prospective study, the characterized retrospective study, and we put together this consensus where we say we're going to call the more than 5 CTC stage 4 aggressive, the other stage 4 indolent. It is true if we have the value and the stratification in the context of every single subtypes and specific metastatic sites. And you see that no matter if you are de novo disease with metastatic disease, a presentation or you have a recurrence, the CTC is stratified into different groups. If you have bone disease only or visceral metastasis, stratified into different groups. So very consistent. And look at this data. I mean, if you are a clinician and you know the data from the first line endocrine therapy CDK4/6, you'll be surprised to see that in fact, if you don't have CTC, this is endocrine therapy only. This is before we had actually the CDK4/6, endocrine therapy alone with less than 5 CTC had a median overall survival of 44 months. So clearly the CTC are a stronger predictor of survival and can identify patients that are more at risk for which resistance has developed. So this is to summarize this part. CTC have a strong prognostic value, can stratify stage 4. The prospective study have demonstrated that chemotherapy is better for endocrine therapy ER positive patient with CTCs, and this can allow us to predict, to design better study based on stratification of CTC. Now, of course, you know, the CTC's prognostic value can also now today be complementary to the cell-free DNA analysis. In CTC, we can look at single cells, cell cluster, but we can also start to look at cell-free DNA mutations and which mutations are associated with a more aggressive disease. So back in 2017-18, we started to look at patients that have received cell-free DNA analysis, and we hypothesized that, in fact, why the cell-free DNA allele frequency and number of mutations is associated with a high tumor burden and increased biology, maybe there is a correlation with CTC. So what's the biology underlying the high CTC? And so we saw this association as well as complementary value, and then we start to look deeper into the molecular analysis of CTC. So when you look at the patient, for example, with estrogen positive disease, and you use the same technology, you can also get some of these cells with a single cell analysis. So you can look at the expression of ER in the 4th channel of this cell search system. Then you can look at the mutation within the CTC. So you start to see heterogeneity. Patients that have CTCs, a component of them are ER positive, and a component of them, not all of them, may have individual variant of ESR mutation. This was back in 2017 when the ESR mutation was just described. It was not investigated in the clinic extensively. We certainly did not have drugs to specific treatment for people with ESR mutant. So as you know, the standard of care for hormone receptor positive is a CDK4/6. And the question now, the number one question is, what do we do after CDK4/6? Should we give the same treatment? So I was involved in the design of the PACE study. That is a study that takes patients that have failed first-line combination with palbociclib and other CDK4/6. And the question that was asked when we designed was, should we go back to single-agent endocrine therapy, assuming that the resistance just to an ASR mutation, for example? Should we just change the endocrine therapy and continue palbociclib, or should we give immunotherapy, checkpoint inhibitor with evolumab? When we designed the study, we want to put together a very good series of correlative studies that include cell-free DNA and also CTC at 2 different time points. So the study was presented, and it's going to be published soon, where it shows that the comparison between group 1 and group 2, there was no much difference. After CDK4/6, if you don't stratify your patient, there is no difference between the combination of fulvestrant. Why there is a difference when you analyze group 1 and group 3. So the 3 drugs seems to be better, even though the small studies was not statistically significant. But when you start to look at the mutations, you see that patients with ESR mutation, they do worse if you do just a fulvestrant. So you start to say that, in fact, stratification is an important factor. So when you look at the CTC in the context of this group, remember, these are post-CDK4/6, there is a good stratification as demonstrated in every single study. And look at this as a proof of concept now in a prospective study where everybody received the same treatment. So the group of patients that receive fulvestrant, the group of patients who receive the blue line on a more than 5 CTC fulvestrant does very badly. So single agent endocrine therapy, second line, if you have more than 5 CTC, this is proven in a prospective study, should not be the choice. So what should be the choice? Well, again, now we start to look at the DNA, get more into the personalized precision medicine that Manny was talking about. Again, I was involved in the design on the steering committee of the Paloma 3 studies. And this was a very heavy in correlative study. We wanted really to know when patients have endocrine resistance and started to get a CDK4/6 with fulvestrant in this case, what's a genomic alteration and the alteration in the tissue that we can predict recurrence, we can predict resistance. And then we took at different time points cell-free DNA to analyze essentially the mutation. What we found out, for example, that the number one with the ESR mutation was 27%, was also polyclonal in 38% of the cases. So these are endocrine resistant aromatase inhibitor for which the ESR mutation is one of the major mechanisms of progression. But you can see it was very clear that when you give the combination, fulvestrant is effective in this ESR mutant, and the combination has the same benefit. So this was the initial analysis based on progression-free survival. Then we measure the cell-free DNA at the time of progression. Imagine that this patient with the combination stay on treatment for longer time, so they are more exposed to endocrine therapy, and the other patient give a shorter time. But the two major mutations that appear to be increased are the PI3K mutation variants and the ESR mutation. And when you start to look at the variants, it is a little bit more sophisticated now. We're not just talking about ESR mutation, we are talking specific variant, they indicate resistance to a specific agent. And I'm telling you this because now there is more and more research on developing agents against ESR mutant and the possibility that in fact different medication, different drug of these SERDs are active. It depends on the variant. So the final analysis of the pallometry demonstrated in fact ESR mutant at baseline or at any time was prognostic. So there we are, CTCs and cell-free DNA with ESR mutation. In the latest analysis integrating both from the PACE study showed that in fact the CTC positive have enriched or ESR mutant in the cell-free DNA. So now we have another explanation of mechanism of resistance why the CTC group is the one group with more aggressive features. So let's assume that in fact in the clinic you have access to this fancy NGS test. This is a report from a Guardian 360 in a patient follow longitudinal. Crazy expensive, don't do it. But just to show you, this is a disease that is characterized by one variant of ESR mutation with a certain allele frequency. The patient was on endocrine therapy with a CDK4/6 with Verzen. You had to interrupt at some point for liver toxicity. And the disease was primarily first in the bone and then eventually in the liver. So when you monitor these patients over time, you start to see that in fact, at the time of progression, there is an increase in the allele frequency of this mutation. You see the images of the cell-free DNA. The disease has changed a little bit. FGFR mutation is associated with endocrine resistance, ESR very high. Clearly, ESR mutation drives this progression. So we did a CTC as well, and she had 12 CTC. So we know by definition it's a stage 4 aggressive with an increased allele frequency ESR mutation, what type of drug you can give. So clearly we have a drug. We have elacestrant that is approved for ESR variant mutation in metastatic disease. But the drug is not extremely effective in patients who have refractory disease with visceral crisis, in which this woman had a very high LFTs. So we started, in fact, with sacituzumab, ER-positive refractory disease. And she had a decrease in allele frequency. You can see very clearly how the ESR variant decreased with the cell-free DNA, and the CTC goes to 0. So we know from what I told you that we are making a difference in this patient survival. At the same time, though, she developed again liver toxicity. We had to discontinue the sacituzumab, went back to elacestrant, but only 3 months later, she had a progression of disease, not shown by the cell-free DNA, but demonstrated from the CTC that went back to 10. Very powerful combination of liquid biopsy to tell us how this disease is behaving. So we have to go back now. This woman was also 1+ HER2, so she's on an HER2. It seems to tolerate it well, and the liver function tests are improving. So going back to the ESR mutant and the medication that the SERDs that we can use in this setting, there is a major effort in trying to develop this drug for the individual variants. In this case, it's lasosoxifene that is better and superior in terms of decline response, reducing the ESR mutant level compared to fulvestrant. You can see the effect on the individual variants now. So this is the level of sophistication that we can get into when we look at the use of cell-free DNA. First, in the trial design, in the trial monitor, in trying to identify which is the best patients in a personalized approach, and second, to obviously use this in the clinic. I mentioned to you the PI3K mutation and the AKT mutations. These are very common. The other possible actionable mutation you can see in a cell-free DNA. this drug capivasertib after alpelisib has been approved based on Capitello studies. They show that after CDK4/6, if you have one of these mutations, certainly can benefit from the combination with fulvestrant and capivasertib. Toxic drug, very expensive drugs, but this is the way of the future, as we were saying. In the molecular tumor board, we discussed which is the best treatment based on the molecular information that you achieve from the cell-free DNA for tissue analysis. Now that you have access, potentially access, to all these drugs that may have shown in clinical trial to improve the progression-free survival of the patients, and probably a trend in overall survival. So I think obviously Mani showed this very clear, that everything you can imagine is real, you make it real. So we need to imagine a moment where liquid biopsy will tell you everything about the disease history, the progression, the evolution. So we started to, again, looking at our large database of 2,000 patients, asking what happened with the definition of subtype at the molecular level, ER positive, triple negative, HER2 positive, when you look at the mutations of cell-free DNA. We saw obviously the most common mutations, PI3K is always present, and the P53 particularly in triple negative. But P53 also defining endocrine resistant, and we just published also present in HER2 ER low patients. So we have then looked at the possibility, as I show you with the Guardian test, that maybe you can actually define response using cell-free DNA criteria. And this is true because we've seen that the tumor burden increased by imaging. You increase the allele frequency of the different mutations that are associated with the disease. And you increase the number of mutations, you also have a more aggressive progression. So all of this, you can put it together and define really some criteria for blood-based evaluation. And then look at the different pathways. If ESR1 and PI3K mutations variants are the most common in metastatic triple negative, can we identify which pathway they are responsible for? So we're getting to a point where using bioinformatic artificial intelligence, you can predict which variant and which mutation can tell you can the disease progress to, what is associated with liver metastasis, lymph node disease, etc. You just in numbers and data in order to be able to do that. And then, you know, because the blood test is more sensitive than imaging, you can imagine that you can predict the progression and intervene before this happened. This was a woman that had been diagnosed with metastatic disease de novo, with lobular cancer. And so once she was found metastatic, she was started on letrozole. So we started to monitor her cell-free DNA. At some point, we noticed that there was an increase. First of all, there was detectable ESR mutation that is associated with resistance to aromatase inhibitor, and the imaging was absolutely unchanged. So knowing that this is an individual that may progress to the liver, what do? You change the drug before this happens. Or at least this is not standard, but it's going to become standard. And the allele frequency decrease, the mutation disappeared, this variant. The patient has been doing well for a long time, then eventually progressed, and she started with another targeted therapy. So this is really monitoring and intervene very early on to maintain a disease that is under control, asymptomatic, and can allow to be lived for a longer time, as Mani was saying. So I think there are several options for post-CDK4/6. Important to know, you already know about the CTC. Now you learn about the ESR PI3K mutations using cell-free DNA. So you can identify and use, if you had access to every drug, every possible drug, based on this concept of molecular progression and of course the patient's conditions and function, you can decide if a targeted therapy is the most appropriate because you have it, because the cell-free DNA is telling you what is appropriate for this patient, so a more aggressive therapy. Where is the cell-free DNA field moving? Minimal residual disease. We talk about the possibility that in fact aggressive triple negative will progress because as a recurrence and the resistance to treatment develop very quickly. I can give you an example because we can talk for hours about this concept. But you know, there are patients that are treated now in neoadjuvant immunotherapy, chemotherapy, and respond very well. And this was a patient with an inflammatory breast cancer, had a complete response except for minimal few cells remaining in the residual lymph node. Complete radiation therapy, continue pembro, adjuvant capsicum, everything you can imagine. So she started to be monitored with a tumor-informed MRD test called Natera or Signatera. And she was found to have, in a short period of time, an increase of this number of molecules as an indication that something was happening. She had immediately a PET CT, so she was not symptomatic, and she was found evidence of lymphnodal disease. So at that moment, she had to start treatment. Of course, the biopsy confirmed was still a triple negative, but you see how important it is to predict and to identify the recurrence before this patient could have been identified later with more symptomatic disease. This was an evolution in the story of the disease, and there are now a number of patients that we have been able to really discover metastatic disease before this evidence of recurrence by imaging or by symptoms. So we know that with a tumor-informed test, the median lead time can be up to 8.9 months. So imagine what can you do in this period of time when the development of disease is already in process. Or imagine if you were to use a combination of molecular tests where you have not only indication that there is a tumor, but also some information about the molecular alteration that you can target before the tumor becomes evident. So there are a number of scenarios for MRD. You can intervene, you can monitor and intervene, you can randomize to different interventions. But I think important is that the field is moving very quickly and try to increase survival by early intervention. And there are also tests that are not only based on genomics and not only based on tumor-informed. These are tumor non-informed with methylation and mutations that I think is going to become very important to monitor patients that are ready, for example, on endocrine therapy, the one that may develop this late recurrence that Manny was referring to. But I think, to me, the most exciting possible application to really affect cancer mortality at a global level is breast cancer screening. We know that breast cancer screening is a challenge for the number of mammograms, for adherence to guidelines. Even the guidelines are not very clear when screening mammogram should be started. So imagine that you can detect the cancer not only at the time of initial diagnosis, a stage 1 disease, but you can detect the cancer before it becomes an invasive cancer, before everything that Manny told you is going to happen and start the process of metastasis. So the survival rate will improve, not only because we detect an early cancer, but because a cancer can be cured by simple modalities, surgery, radiation, and simple and non-expensive treatments. So this is the only way to do it. And of course, there are a number of proposed technology. One of them, of course, is a methylation test, multi-omics, but also looking at multiple tumor detections, not only breast cancer. Based on the methylation profile, you can potentially detect different tumor types. And actually, breast cancer is not even the number one example, because detecting stage 1 breast cancer in this particular study published in 2021 with a methylation assay, the GRAIL study, is not really very effective. But there is a study looking at the Pathfinder study, looking at an elderly population. I need to be careful when I say elderly. An older population where there is an increased incidence of cancer, and potentially we can discover this cancer at an early stage. Obviously, the number of screening is very high because it happened that the detection rate has been very low, as possibly expected. So you need to screen many patients, and this study is designed in a way that is not feasible, doing a PET scan after the positivity and then follow those patients. There is the Siantra diagnosis test, for which I got a DoD grant for patients that have undergoing mammogram, biopsy, recall, high risk by genetic risk, young women for which mammogram is not even indicated. If we can identify the signal positive and proceed with biopsy, there is a concordance, obviously we have a positive test. This can become more standard of care, not to replace mammogram, but to integrate a complement. And then, of course, there is a local test potentially from the TAR, where we have the ability to detect cancer cells instead of DNA. And if you can do this in a very early stage, you can also achieve the same goal. So this is a summary of how the cell-free DNA and the CTC and in the future after blood-based diagnostic can inform about the disease natural history at any time. Early detection, monitor during treatment, particularly neoadjuvant treatment, MRD, molecular recurrence, molecular progression, and of course resistance in the context of every advanced condition. And the MRD can be tumor-informed or not, and the tumor-informed is important probably for the first 5 years. Late recurrence has to be non-tumor-informed based on methylation. So this is a quick summary of what liquid biopsy can do. It has been my personal journey from the beginning with the CTC. This is just a summary that CTC and cell-free DNA detection are increasingly used in clinical management. Hopefully, we hope the cost will be reduced because really gives a good view what the disease is about. At any time point, is a real-time assessment of the disease and can be used for longitudinal studies during therapy, decide when to treat the disease, when to change, and in the future, as I mentioned, is tumor interception. Can you change the trajectory of a cell that is becoming invasive cancer? I don't have a large lab because I'm primarily a clinician translational research, but I do have 9 people working with me, primarily on CTC, cell-free DNA, and we have a large collaboration with other investigators in other institutions. And of course, always happy to work with Mani. We also work with different companies, receive some grants, and I just want to remind everyone there are only 2 ways to live your life. One is to nothing is a miracle, and the other one, I think Mani showed very clear, everything is a miracle. Thank you.
If we have any questions, audience can kindly raise their hands.
Massimo, thank you for a beautiful talk. You talked about actionable mutation in a ctDNA and a CTC. Now, both are in the liquid biopsy space. Do you think they both characterizing CTC and ctDNA would be complementing, or would they be like one should do both, or just one would inform for classifying the patients better?
Yeah, so obviously this is a question of multiple implications because as we talk about, I showed a very clear example where you have a cell-free DNA with a very good sensitivity that is able to detect variant of mutations. This is not always the case. Sometimes the tumor is not shedding, for example. So you still need a tissue biopsy for that purpose. Now the CTC are complementary. So I think the CTC is more representative of the metastatic process, and probably if you were to have a fast, cost-effective analysis of CTC mutation, that would be the way to go. We know that the cell search is not the ideal system because it is only capturing a component of the cells that are not EMT or not completely EMT. So we are losing some of the cells that may be informative, especially for triple-negative disease. But imagine if we had a very good, not enrichment, but not immunomagnetic base for CTC to be addressed and evaluated in the variability of population that we have. And we had a chance to do in a very short time mutations, like in a week, because once we talk about this test and we talk about this analysis, we also put in the context of treatment planning, how long it's going to take to do some specific analysis in order to be able to treat the patient as fast as we can. So yes, they are complementary.
Thank you.
Thank you for the wonderful talk, sir. My question was, right now, are there any treatment modalities that focus on preventing metastasis of cancer by preventing the cellular invasion into blood and lymph vessels? And would such a treatment be useful?
Yes. So we don't have this type of treatment. I think we assume as oncologists that we treat in a very empiric way, I would say. The treatment in the adjuvant setting are supposed to prevent metastasis, even though we know this spreading of cells has already happened. And they focus on basic characteristic of the patient, endocrine sensitivity, chemotherapy. So none of the therapy has been developed to prevent metastasis because, as Mani already explained, if you assume that the metastasis is due to the stem cells, that these are only a fraction of the cells, we don't have a good way to treat. For how long we treat, which is the best setting? Probably the adjuvant setting, or maybe it's the neoadjuvant where you eradicate the cells before they can even reach other organs. So there is not, but there's been a lot of search for that. You know, when they started to look, for example, disseminated tumor cells in the bone marrow, and not CTC, before CTCs, there were, you know, the bisphosphonates and these other drugs that were developed to affect that population of cells. The cells that were already out were found to be associated with the metastasis to prevent the metastasis. The CXCR4 inhibitors, mobilization of cells from the bone marrow. Nothing has been approved, nothing is used in the clinic, so we definitely need to refocus and try to go back and see if we can actually work on this medication. But the field is, unfortunately, is very rich in type of treatment based on what is already established and not necessary to prevent what can happen.
Thank you, sir.
Excellent talk, sir. So one question, this was metastatic. If we are, I'm just thinking whether we are dealing with a non-metastatic cancer, we have given the treatment, PET scan done, follow-up, nothing is there. We do a circulating tumor cell, just adding to our question just to know whether, and we find there are circulating tumor cells, but imaging wise, clinical wise, there is no disease. Whether in such a patient we consider some maintenance therapy, we still go with continuation of adjuvant, or is there any data regarding that, or we just observe?
Yeah, so with regard to the CTC, as you know, there were several studies in Europe, particularly in Germany, that showed that you have CTCs, after you finish adjuvant treatment, these patients are at risk of recurrence, develop recurrence. There was never a follow-up for intervening. There was only one study that were looking at the CTC in the adjuvant setting that are also HER2 expression, but that because you have a smaller proportion of patients that have CTC and a smaller proportion of patients that have HER2 positive, that study could not be finalized and never closed. So the idea is not only to find CTC, but to try a therapy that will be effective on that. So the answer is no, but the same concept can apply to MRD when you have a detection of tumor. Right now, I think we are getting in a situation where what if you are MRD detection in someone that has already received a CDK4/6? If you don't have a CDK4/6, you may consider to start this in combination with endocrine therapy as part of your adjuvant, but also early metastatic. But if you have already been exposed to CDK4/6, what do you do? And I think all these questions can be addressed with some of the prospective studies, some of the retrospective real world. It's going to be a very complicated field in the near future, because some of this medication will need to be moved very early on. But fundamentally, if you want to use a liquid biopsy, I think what we need to go is to start measuring at the time of neoadjuvant, because you want to know if that residual disease was there, was detectable, and would be gone by your treatment. If you measure only afterwards, it could be residual disease. I didn't have time to look at some examples that we have in the clinic from the fact that you had a locally advanced tumor, just never completely eradicated. Same concept that you have a pathological response.
Thank you very much.
It was wonderful talk, sir. I have a question, like these CTCs are very low in number, so it's chance to miss out, like, so is there any like way to like enrich them or something like that in the same form? Like?
Yeah, so the CTC are very low with the cell search system. When you use a microfluidic system and filtration system, you see a much larger number of cells. So it depends on what's the question, what's the setting. I think you can probably select which technology you're using. We have parsortic system. We have been working on actually the filtration. I didn't have time to show as the best to identify clusters, not only single cells, in early disease. There is more clusters in non-metastatic patients. And the question is, if the clusters don't go away through treatment, is this the setup for metastasis later on? Well, in that case, you don't use the cell search, use the filtration system.
Okay. Thank you.
Sir, my doubt was in normal regimen that we use in liquid biopsy, blood is of course the most important, but in normal regimen while detecting, do we also check other body fluids like CSF and lymph? at the simultaneously, or do we check only blood and then if indicated we check in other samples? And if we do, then what is the, like, is there a big difference in the detection rate of cancer cells?
Yeah, so that's also an important question. I think the blood is certainly the most important source, but you can also see a difference between venous blood and arterial blood, and there have been studies looking at that for lung cancer patients or for colorectal cancer at the time of surgery, where the arterial blood always had more CTCs compared to the peripheral vein. In regard to the CSF, there are patients with leptomeningeal disease where we can do CTCs. We can do CTC with biomarker. One of the questions now is a patient they have, for example, a triple negative risk of high development of brain metastasis. Can we have a technology that can detect these patients? And we are studying also in our institution a combination of CTC, cfDNA with different methods to see if this brain metastasis can be identified earlier. The other group will be the HER2 positive, complete response to the rest of the body but develop brain metastasis. And if you know, brain metastasis is a very challenging. There was one study that was never published that showed actually the CSF, cfDNA, after you complete treatment, was able to identify patients who develop brain metastasis later on. So it's MRD in the CSF in a way. But that's obviously it's an invasive procedure, and I don't know if this is going to be done prospectively for future studies.
Sir, why don't our immunity suppresses these CTCs when they are circulating through the blood?
I'm sorry?
So why don't our immunity suppresses these CTCs?
Yeah, we haven't studied the immune contribution of that. There are very few studies that look at flow cytometry to define the T cells and the CTC at the same time. So there is been minimal, if any, confirmation that in fact an immunosuppressive environment associated with higher CTC is possibly the case. But in a way, I told you that when you have CTC, you have more mutations, so it's a more aggressive tumor that evades the immune system. So that's probably what happens if you were to have a group of patients where you have an immune deficiency, not real immune deficiency, but immune cytotoxic deficiency.
Sir, is it possible,
Yeah.
during the initial stages of cancer, is it possible to know which tumor is going to be metastatic during the initial stages when it has not spread?
Yeah, so typically we do imaging to see if there is any established metastasis, but if you were to do CTC, you can predict which patient will have a higher chance to develop metastasis. The flip coin of this is also that there are studies that were trying to de-escalate treatment. For example, there is a study we're looking at de-escalate radiation therapy in patients that don't have CTCs when they have a very small tumor. Obviously, every treatment modality has side effects and some costs. So if you don't have an indication of local distant recurrence, maybe you can reduce the use of radiation in this population at lower risk. So you can de-escalate or escalate. Even that, I think, is a good use of predictive prognostic.
So if it's possible, then we can treat the metastasis before actually it begins spreading. I mean, so that's why.
I think that's a great plan for the future. Dr. Mani is working on a drug that is really affecting metastasis. So if we can give a non-toxic treatment at an early stage of the disease, as I say, I think that would be ideal.
Thank you, sir.
Sir, cell invasion starts because a cell loses their signaling property. So can you explain me something about why cells lose their signaling property?
I can't.
Metastasis occurs because cells lose their cell signaling property, and the signals which they take from stem cells that when to grow, when to stop growing, these signals lose there. So can you explain why they lose such signals receiving from, or they do not obey such signals?
Mani, you can try to.
Online. So I will answer that. The cancer cells, they're trying to find a way to escape from one place. The reason is, imagine this room, There is no place like, everybody is kind of overcrowded, right? You will find a way to go to your room and use a laptop to listen to the same talk rather than sitting in a crowded space, right? So same way, tumor cells are finding a way to leave the primary site wherever they are when it's crowded. So when they leave, they lose the cell-cell communication, but they don't lose the signaling, right? So the signaling, there's two kinds of signaling. Me telling the person sitting next to the person, communication. The second communication is there is communication going on in my brain to myself. I'm just saying, okay, now should I sit here? Should I go there? Should I eat this? Right? That's a constant communication, even though it's internal, but there's a communication, right? This communication never goes away. That communication goes away to the neighbor, right? And when they go to the newer site, they create a new communication to the newer place with the newer cells. And the new environment say, hey, 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 you don't belong here. Don't come to my room. Go to your room, right? So things like that. Make sense?
Another way to say, for example, we know that cancer is based on mutations, change in oncogenes. There are some mutations that lose suppressive gene capabilities, and so these cells are more aggressive. Their ability to survive and to escape. So from a genomic point of view, there are mutations that predispose cancer to lose that signal, to migrate and to create metastasis. I think P53 is an obvious example that comes to mind.
One more question was this, sir.
We can obviously interact post this session as well. For now, given the time constraints, we will conclude the session. Thank you, sir. Thank you, everyone. Thank you, everyone, for your attention. And I'm going to steal this moment just for a personal reference to explain how excited and honored I am to compare this session today. It's because one of my first review articles which I published back as a MSc dissertation student, while I came to know about the profiles of the speakers that we would be hosting, I am so excited to say that I have cited the best of researchers, Dr. Mani and Dr. Massimo, both in my review article 10 years ago. And that's the reason it's my favorite, because today I know I had cited the best back then. So thank you again, sir. Thanks to everyone. I have been Dr. Navjeet Kaur, research coordinator from CIRAC, and it has been an honor and pleasure comparing this session. And I would now like to invite Dr. Moshmi from CIRAC to present the vote of thanks. Thank you, everyone.
Dear ladies and gentlemen, a very good afternoon. Before I go into my formal vote of thanks, there is something which is very live thought in me. And as you talked about your TED Talk, I was Google about your TED Talk maybe 2 days prior, and I was sure you will need a collar mic today. But we are very sorry about that there was a glitch about that collar mic, but I was very sure you wanted to take this stage and express yourself in a very expressive manner. Now, going ahead with this, another thought which is coming in my mind is like, you both have actually covered the most updated status of cancer research which is happening across the globe right now. And you both appear to me at right this moment as a cancer Wikipedia. Cancer Wikipedia in cancer research, most updated. You have covered cell-cell signaling. You have covered tumor microenvironment. You have covered diagnostic. I was looking forward a little bit about immunodiagnostics, which even this young fellow, he asked. Immunodiagnostic, of course, it is too challenging. And this cancer research on whole is a very challenging research topic. Though the young minds are asking you several questions, these are really complex and complicated disease. This is research part, and there are many of us which have actually seen our family members and our friends which have actually undergone these type of stress. And I would say with patients, clinicians like Dr. Massimo, and the close family and friends, they are the ones which are actually experiencing what is happening with the patients in the later stage. So now going with my formal speech, while thanking you all of us who has gathered today, distinguished guests, dignitaries of the dais, senior and fellow colleagues, participants from Somaiya Vidya Vihara family, and beloved students. I am Moshmi Kumar. I am Associate Professor at Somaiya Institute for Research and Consultancy, and today it is my honor and privilege to give a vote of thanks to all those who made us in this gathering today and bringing us all together on the platform of Somaiya Public Lecture, which is an ongoing endeavor at SVU. I take this opportunity to express our gratitude to the brilliant minds, Dr. Sendurai Mani and Dr. Massimo, who have shared their experience with us today. Your contributions have shed light on the critical aspects of cancer biology, its diagnosis and treatment strategies, which contributes to our collective journey towards understanding and combating this complex disease. While fighting cancer, families have overcome what seems impossible. Cancer cannot cripple love, it cannot shatter hope, it cannot conquer the spirit. It got reinforced once again with Dr. Mani's and Dr. Massimo's talk, where we could see that diverse perspectives have converged, as well as clinical and cancer research collaborations have thrived. On behalf of SIRAC, Somaiya Vidya Vihara University, I am deeply grateful to our prestigious sister organizations under the Somaiya Group flagship, having multiple settings involving participants from diverse backgrounds, attendees from Sajjan Therapeutics, which is a state-of-the-art R&D facility with expertise in anti-cancer drug design and development, making them a strong player in the fight against cancer, and medical professionals from KJ Somaiya Medical College for their unwavering support in making this session possible. A special mention to our Honorable Chancellor, Shri Samir Somaiya, for his visionary leadership and dedication towards academic and translational excellence in the field of cancer. My gratitude to our Honorable Vice Chancellor Professor V.N. Rajshekharan Pillai for his continued support, the Registrar Office, the PR team, and my fellow colleagues under guidance of Director Dr. Achla Danayat and Dean Research at CIRAC for their timeless efforts in organizing and promoting these illuminating sessions. Heartfelt thanks to each participant for carving out time from your busy schedule and making it to the Somaiya Public Lecture today, contributing to the advancing of the knowledge in the scientific research and innovation. I would also like to thank our event organizer and campus coordination team, along with student volunteers who have helped us and make this session possible and helping us in setting up the event. As we disperse, let us carry forward the knowledge and inspiration gained from these sessions into our respective fields of study and research. Together, let us continue to strive towards the advancements in cancer research for researchers and patient care for clinicians and close family and friends. Once again, my genuine thanks to all of you for your invaluable contribution and support. Enjoy your day and have a happy weekend. Thank you so much.